The motor neurons were designed in a way that they have the potential to communicate with what we call the skeletal muscles. They communicate through a very beautiful space whereby there's going to be release of what we call a neurotransmitter, acetylcholine, that's going to be stimulating the muscle fiber leading to skeletal muscle contraction. This union between the motor neurons and then the skeletal muscle fibers is what we call the neuromuscular junction. Good morning to you all. My name is Tumnad, and in today's video, we are going to be focusing on what we call the neuromuscular junction. We shall look at the transmission side, we shall look at even the physiology of it, and then we shall look at some of the little of the clinical correlation. Okay, so now, but if it is your very first time to watching, please make sure you first subscribe, you turn on notification bell so that whenever I load the video, you become the very first person to watch. So let's go ahead and get started and look at what we call the neuromuscular junction. Okay. Now, here what you are seeing is a diagrammatic illustration showing you that we are going to be seeing a communication between the neurons and then the muscles. Now, what you need to know that when we say neuromuscular junction, it means we are going to be having two kinds of cells communicating to one another. We have a neuron, and then we also have muscles. Okay, those are the two key important things to note. We have what we call the neuron. We have what we call the neuron, and then at the same time we also have what we call the muscles. And to be even more specific, we are going to be looking at what we call the skeletal muscles. Okay, we are going to be talking about the skeletal muscles. And when we talk about the neurons, they are going to be motor. Okay, they are going to be motor neurons, and even to be even more specific, is going to be under somatic nervous system. Okay, they are going to be what we call somatic. And when we talk of somatic, in the mind you have to think of what we call the skeletal muscles, and of course skeletal muscles being under what we call voluntary control. Okay, when we talk of somatic, we mean skeletal muscles, and of course which are under what we call voluntary control. Okay, so now these are the key important things when we are talking about the neuromuscular junction. It is the neurons, which is somatic motor or somatomotor neuron, which are going to be communicating with what we call the skeletal muscles, forming what we call the neuromuscular junction. Okay, so now here we are going to be looking at this diagrammatic illustration. We see how the these motor neurons are going to be communicating with the muscles. Okay, now we need to first know this is a cross section. We want to know where are the cell bodies of the somatic motor neurons going to be coming out from. This is going to be now our cross-section of the spinal cord. These are going to be our posterior horns, or what we call the dorsal horns, and these are the anterior horns, which we call the ventral horns. In the ventral horns, that's where we're going to be having the cell bodies for these somatic motor neurons coming out, meaning that this is going to be now the axon of the somatic neuron. Okay, so now this is going to be now our motor. This is going to be now our motor neuron. Okay, this is going to be our motor neuron. Now, our motor neuron is going to be terminating, it's going to be making multiple of what we call the axon terminals. But we have zoomed out on one, and we want to see how it is going to be communicating with this kind of the muscle, with this kind of these muscle fibers. We're going to be seeing multiple axon terminals, but we have just cut on a very small piece of one axon terminal with this muscle fiber, and we have brought in here, we have zoomed it to this point here. Okay, this kind of illustration is what we have zoomed and we have brought it here. Let us see how these motor neurons are going to be communicating with what we call these muscle fibers. Okay, and of course at the back of the mind, that's going to be skeletal muscles. Okay, now we need to first know how we call all these different structures of this kind of neuromuscular junction. The whole of this kind of this bulbar structure, the whole of it is what we call the axon terminal, or even may call the terminal endings. Okay, we're going to be having a lot of different structures within, but you see these channels, these red channels, they are voltage-gated calcium channels. Okay, they are called voltage-gated calcium channels. Now, you see these vesicles. Inside these vesicles, they are what we call the synaptic vesicles, and they are going to be housing one of the most important neurotransmitter in this neuromuscular junction called acetylcholine. Okay. And then this area which is going to be facing our muscle fiber, this membrane is what we call presynaptic membrane. okay? And it's going to be having on the inner surface, going to be having these kind of snare proteins, okay? You have this what called snare proteins. Even the snare proteins are also going to be on the membranes of these vesicles, okay? So now the snare proteins which are on the presynaptic membrane, they are what we call the T snares. We have the SNAP25 and then called Contraction, okay? And then the snare proteins on the vesicles of these synaptic vesicles, we have what we call mainly what we call synaptobravin, and may find what we call synaptotagmin, okay? And then also going to be having the powerhouse of this cell, which we call the mitochondria, okay? And now the whole of this space, which is around close to 30 nanometers, very small microscopic space through which the neurotransmitters are going to be powered, is what we call the synaptic cleft, okay? Now, when you see this kind of the snake, it is a very beautiful enzyme, and this enzyme is responsible for breaking down the neurotransmitter acetylcholine after the electrical impulse has crossed the synapse. This enzyme is what we call acetylcholine sterase enzyme, responsible to break acetylcholine. Okay, now this area of this axon terminal, where it is going to be synapsing area, that exact area of this muscle fiber is what we call the motor end plate, okay? And of course, the motor end plate is going to be having these kind of foldings, okay? These kind of foldings are going to be increasing the surface area for the action of what we call the acetylcholine, okay? Now, we need to first understand how do we call these kind of invaginations, okay? These kind of invaginations are what we call the subneural clefts, okay? They're what we call the subneural clefts. We may even find some people calling it post-junction folds, okay? They have a couple of names, okay? They are going to be having these kind of protein channels. You see these kind of protein channels, they are pentameric proteins, which are having 5 subunits, the 2 alpha, the gamma, the beta, and then the delta. These channels are super very important. They are called acetylcholine nicotinic receptors. They are going to be more of ion channels, such that when acetylcholine binds there, they open and allow cation to move in and out. Okay, for that reason, they are even more of ligand-gated ion channels. It is such another beautiful thing. Okay. Now, we also have these proteins which are going to be very important on the presynaptic membrane. These proteins are super important in the way that they have the ability to recycle these vesicles. They are called clathrin proteins, okay? They are called clathrin proteins because we shall see that in the case of neuromuscular junction, you are going to look at that the acetylcholine is not going to be made in the cell body. It's going to be made in the axon terminal. Okay, we are going to be looking at that on how acetylcholine is going to be made. Okay, so now having looked at that, so let's go ahead and see how neuromuscular junction transmission occurs. Okay, but at the back of your mind is that for anything to happen here, like the release of a neurotransmitter, any physiological process which happens in the axon terminal, we need to have an electrical signal coming towards it. Okay, we need to have an electrical, or what we call the action potential, arriving at the axon terminal. And of course, the action potential is going to be coming in what we call, you have these channels. These channels are what we call the voltage gated sodium channels. They are the ones which are going to be helping us in the movement of this kind of the positive ions, or what we call the depolarization wave. Okay, so now when an action potential arrives at the axon terminal, what is going to be happening? These voltage gated calcium channels are going to open. Okay, when they open, they are going to be allowing the influx of what we call calcium. Calcium is going to start entering within this this axon terminal. Now, what is going to be happening? When calcium enters here, these vesicles are going to be containing acetylcholine. Now, when this calcium enters here, we have these snare proteins like the syntaxin and then synaptobravin and then SNAP25. These kind of snare proteins, they are going to be wrapping to one another by the effect of what we call calcium. And in that way, these synaptic vesicles are going to be pulled until they fuse with what we call the presynaptic membrane. And when they fuse with the presynaptic membrane, then you're going to be seeing our neurotransmitter getting powered into the synaptic cleft. And this neurotransmitter is what we call acetylcholine. Now, When acetylcholine diffuses through this synaptic cleft, it's going to come on the motor end plate and bind on a specific receptor on what we call these kind of nicotinic receptors. Now, when this acetylcholine binds on this acetylcholine nicotinic receptor, these channels are going to open. Okay, these channels are going to open. Now, when they open, two things happens. We are going to be having movement of cations, and here we mean sodium and then potassium mainly. What happens is that when sodium enters, then potassium exits. But here we have to be very cautious in the way that there is going to be more of the sodium influx than the efflux of potassium. And this is going to make a very local kind of depolarization going to be generated inside here. And that kind of local kind of depolarization which is going to be generated is what we call the end plate potential. Okay, is what we call the end plate potential. Now, it is the strength of the end plate potential that will lead into what we call the generation of an action potential that will flow around the muscle fiber membrane, which will lead into what we call the skeletal muscle contraction. Okay, after the skeletal muscles have contracted, we need these kind of neurotransmitters to get broken down, because if they stay here, they're going to continuously keep stimulating these muscle fibers, leading into what we call continuous contraction. That's why we have this kind of enzyme called acetylcholine sterase enzyme that keeps breaking acetylcholine. Okay. So now that is briefly how neuromuscular junction happens. Let me just recap. Arrival of an action potential leads to the opening of voltage-gated calcium channels. Once calcium enters into this synaptic knob, it will lead into the fusion of what we call the synaptic vesicles containing acetylcholine, and eventually release acetylcholine into the synaptic cleft. When acetyl is released into the synaptic cleft, it diffuses and then binds on a specific receptor called nicotinic receptors, which will lead into the influx of the sodium ions and then efflux of potassium ions. But because the sodium ions influx is even very, very much more than the potassium efflux, it will lead into what we call a local depolarization, which we call the end plate potential. If the end plate potential is sufficient enough, it will lead into the generation of an action potential, which will run through what we call the sarcolemma and to lead into what we call skeletal muscle contraction. We shall go into deep and we see how this kind of the depolarization will go and lead into the release of calcium from what we call the sarcoplasmic reticulum. Okay, but before we go into that, we want to first look at what we call the synthesis of acetylcholine, because we have realized that this acetylcholine is going to be one of the best neurotransmitter in this case. Now, what is acetylcholine? Acetylcholine is what we call acetylcholine is a neurotransmitter for the case of this neuromuscular junction. But acetylcholine is not a protein. What do I mean by this? If it is not a protein, that means its synthesis area, the area where it's going to be manufactured, is going to be the axon terminal, not the cell body. Okay? Because the cell bodies, we are going to be having the DNA that's going to be transcribed into messenger RNA, then messenger RNA leaves, it goes to get translated by the ribosomes to make a protein, and this protein, they can move. So now, those other neurotransmitters, which are proteins, they are going to be made in the cell bodies in what we call the cell bodies. But if acetylcholine, the fact that it is not a protein, its point of synthesis, or the area where it's going to be manufactured, is going to be what we call the axon terminal. Okay, and this acetylcholine is having a very, very, very small molecular weight. Okay, very small molecular weight. It is something which is very, very, very light. Okay, so now what are the two components that make what we call acetylcholine? Acetylcholine is going to be having two components. The first one is going to be what we call acetate, and another one is going to be what we call the choline. Okay, those are the two components that make up acetylcholine. Now, this one may even call it acetyl-CoA. acetyl-CoA. And of course, who is going to be providing us acetyl-CoA? This powerhouse of the cell, which we call the mitochondria, okay? Because this is going to be one of the intermediate process of what we call the Krebs cycle, okay? And then the choline, this is going to be just a nutrient, okay? This is going to be just a nutrient which you can just absorb through the GIT. Okay, when we eat food, we may be having a nutrient called the choline, which we can absorb through the GIT. Okay, so now when this acetylcholine reacts, of course, now on the axon terminal, we are going to be having specific kind of these channels. Okay, we're going to be having specific kind of these channels which are going to be allowing the entrance of what we call choline. Okay, because our acetyl-CoA is going to be inside, but we need now the choline which is going to be coming out from the extracellular fluid. So we need to have these channels that allow the inflow of what we call the choline. Okay, so now when acetyl-CoA binds with what we call the choline, when they bind together, they form what we call our neurotransmitter, which we call acetylcholine. But of course, this process is going to be aided by this beautiful enzyme, and this enzyme is what we call choline acetyltransferase. Okay, choline acetyltransferase. This is an enzyme which is going to drive the process of acetyl-CoA, which is going to be reacting with choline to make what we call acetylcholine, and then they release what we call the CoA. That is something which is very important, and that process is going to be happening inside the axon terminal. It does not occur in what we call the cell bodies of this neuron. Okay, so now having seen that, let's go ahead and see the physiology behind how these skeletal muscles fiber are going to be stimulated, leading into the process of what we call the sliding filament theory. Okay, we said that when a neurotransmitter acetylcholine binds on these nicotinic receptors, they are going to open, leading into more and more influx of sodium and then efflux of potassium. But because we said that there is going to be more influx of sodium than the potassium, it's going to create a very small kind of depression. The reason why there's going to be more of the sodium influx is because the concentration gradient, we have a lot of sodium in the extracellular environment than in the intracellular environment. Okay, so now that's going to be one of the driving factors. And besides that, we have a lot of negative charged ions inside the cell. Okay, and of course that's going to be also creating that kind of pulling force. Okay, a positive is going to be pulled to a negative. So now those are two kind of driving forces increase more sodium influx inside here than the exit of what we call the potassium. Okay, it is also such another beautiful thing. So now what happens? This kind of the local depolarization is what we call the end plate potential. Okay, you may find it even written what we call the EPP. Now, what happens when the EPP, if the EPP is strong enough, it will lead into what we call the generation of an action potential. Previously, this muscle fiber was at rest. It means that it is even having what we call the resting membrane potential. Resting membrane potential is that potential difference, is that charge difference when a cell is at rest. Okay, and mainly it is highly negative intracellularly than extracellularly when at rest. So now, if I draw this graph like this, here we are going to be having time. Here we are going to be having time. Here we are going to be having potential difference in what we call the millivolts. Okay, so now all cells in the body, they have what we call resting membrane potential, but not all cells in the body are excitable. We only have two types of cells that are excitable: the neurons and then the muscles. Okay, so now we are going to be having what we call the resting membrane potential. Resting membrane potential. Now, the point is, what is the resting membrane potential of these skeletal muscles? It is generally close to around -85 or 90. Okay, around -85 or 90 millivolts. That is now going to be our resting membrane potential. Now, what is going to be happening? So now, the end plate potential, which is going to be generated here, it is going to be pulling this kind of the cell from resting membrane potential above, slightly above. And if this end plate potential is super enough, reach what we call the threshold potential, which you may call the TP. Okay, and it is at the threshold potential whereby we are going to be having other kind of channels opening. And what's going to be now our threshold potential for the case of these skeletal muscles? It is going to be generally negative 55 millivolts. That's when our threshold is going to be reached. Now, when the threshold potential is reached, what's going to be happening? Voltage-gated sodium channels are going to open. And when they open, a lot of sodium enters in. And this kind of movement moves up. Okay, you're going to be having now this kind of depolarization. Okay? And of course, this kind of depolarization now can go even up, for example, up to around positive 30. Okay, when it reaches positive 30 millivolts, there are a lot of other processes which are going to be happening. What happens is that now we are going to be having this kind of the sodium, the voltage-gated sodium channels are going to start closing some of them. And of course, at this point, you're going to be having now the voltage-gated potassium channels trying to open. And when they open, you're going to be having now the efflux of potassium, making the internal environment of the cell to become more and more negative. When it becomes more and more negative, this is what we call repolarization. It can go until it comes back to the resting membrane potential around negative 90 millivolts. Okay, so now let us now bring this and we put it here. Okay, now our end plate potential was sufficient enough, and it has caused it has reached what we call the threshold potential. What happens? You're going to be having these kind of voltage-gated sodium channels are going to be opening. Okay, these voltage-gated sodium channels are going to be opening. When they open, you're going to be having sodium influx. So we shall have a lot of sodium coming inside this cell. Okay, it means this area is super now depolarized extensively. Now, these kind of the positive charges of these sodium ions are going to be moving. Okay, are going to be moving, and of course, that kind of way of movement of the positive charge of the wave of depolarization is what we call the action potential. Okay, so now it will move and move and move. You see this invagination on this muscle fiber. This kind of invagination is what we call the T-tubule. Okay, you may even call it the transverse tubule. It is super very important. That's going to be taking the depolarization deep into this muscle fiber. Okay, now what happens is that this kind of depolarization is going to be moving along with this now. These T-tubule, they have super very important kind of these receptors. These receptors. These receptors are called the hydropyridine. Okay, the hydropyridine receptors. Okay, they are called the hydropyridine receptors. And these receptors, they are going to be sensing this kind of depolarization. Okay, and then you see this structure which is going to be on the side of this T-tubule. You see this structure, we have this structure and this structure. These two structures, they are going to be on the sides of what we call the T-tubule. These structures are what we call the sarcoplasmic reticulum. They have too much concentration of calcium ions. These are going to be the factories of what we call calcium. But now, when this kind of wave of depolarization reaches on these receptors, see these receptors, even on the sarcoplasmic reticulum, you see this kind of invagination, these bulbous structures. These bulbous structures are what we call the terminal cisterns. Okay, these terminal cisterns, they are going to be having specific type of receptors, and these receptors which are going to be on the terminal cisterns are what we call ryanodine. Okay, they are called ryanodine receptors, and if you want to be even more specific, it's going to be ryanodine receptor type 1. So now you see this, the hydropyridine and then ryanodine receptors are going to be now interconnected to one another. Okay, so now when this kind of depolarization reaches the hydropyridine receptor, it is going to be stimulating the receptor and it will start pulling on the ryanodine receptors. Okay, when that one happens, guess what's going to be happening? Through what we call the terminal cisterns, you're going to be having now the efflux we're going to be having the out movement of this molecule we are calling calcium. So now, calcium is going to start coming out of what we call the sarcoplasmic reticulum. Now, if we have this kind of the terminal systems with this kind of the tubule, then terminal systems, or those structures collectively are what we call the triad. Okay? Now, what's going to be happening now? Once this calcium is released, this calcium is going to go and now into the cytoplasm, where it's going to go and interact with what we call contractile proteins, like so what we call the troponin and then tropomyosin. But primarily it's going to be now binding to what we call troponin C. Okay, now when this calcium binds to the troponin, it will cause now the tropomyosin to expose what we call the myosin heads. And of course, what's going to be happening, we're going to be having formation of the cross bridges. Finally, we shall lead into what we call the process of muscle contraction, which we call the sliding filament theory. And now, of course, now after the muscles have contracted, we need this calcium to go back into the sarcoplasmic reticulum. If this calcium stays in the cytoplasm, it will keep triggering the process of what we call contraction, which we don't want, okay? So that means after the contraction, okay, we need this calcium to start flowing back into the sarcoplasmic reticulum. You see this sarcoplasmic reticulum, they have these beautiful channels, and these channels are what we call, they are the sucker pump, okay? And these sucker pumps, they are going to be allowing the inflow of calcium ions after the process of muscle contraction, okay? Now, what happens is that because calcium is going to be less in the cytoplasm compared to the sarcoplasmic reticulum, that means that it is going to be moving uphill. It is going to be moving against the concentration gradient. It means these sucker pumps, they are going to be using energy in form of ATP, okay? We shall have hydrolysis of ATP, which will lead into the formation of what we call the ADP, and then also what we call inorganic phosphate. So now, this kind of movement of calcium ions through the sucker pump to the sarcoplasmic reticulum is an active process, okay? And then we have said that after the contraction, when the axon potens, so you see, axon potens is going to be now the depolarization wave, and then of course we have a repolarization phase. That means these muscles, we don't want them to stay in a contracted form. Time has to reach and they have to relax. And how do they relax? We have these channels. See these channels? These channels are voltage gated potassium channels. What happens? They are going to be starting allowing the efflux of what we call potassium, okay? What we call potassium. Now, when potassium leaks out, it means now we are going to be having now this area inside to become even more and more negative, okay? Now, when the inside of the cell becomes more negative, we say it is becoming repolarized, okay? And that's what's going to be pulling this kind of the membrane back, the resting membrane potential. Because we don't need these muscles to stay in a contracted form. They have to relax. So now, having seen all that, let's go ahead and see what we call some of the clinical correlation with what we call the neuromuscular junction, okay? Clinical correlation with what we call the neuromuscular junction. It is also worth to be known, okay? It is something worth to be known. So talking about what we call clinical correlation with what we call the neuromuscular junction, we have a lot of problems that are going to be occurring on what we call the neuromuscular junction. We have what we call this bacteria is called, it is a gram-positive bacteria, anaerobic bacteria called the Clostridium botulinum. That kind of the bacteria, it is going to be producing some kind of the neurotoxic substances. It can release substances like what we call botulinum. Okay, botulinum. Now, these kind of toxins, botulinum toxins, what we call botulinum toxins. Now, these toxins, they are super very dangerous in what we call neuromuscular transmission. You know what these botulinum toxins, what they do? They come here and break these snare proteins. You see these kind of snare proteins, we have what called the syntaxin, the synaptobravin, the synap25. They are helping in the process of what we call the fusion of these vesicles containing the neurotransmitters. So now, if you have these kind of botulinum toxins breaking these kind of snare proteins, That means we're going to be having a reduced fusion of what we call these vesicles containing acetylcholine. That means we're going to be having less of what we call release of acetylcholine into the synaptic cleft. Okay, so now that is also going to be now one of the most dangerous thing which is going to be happening. It will lead into what we call paralysis. Okay, what we call muscle paralysis, muscle weakness, and that kind of the condition just what we call botulism. Okay, what we call the botulism. So now that's one of the most important clinical correlation which you have to be knowing. Then again, we even have an instance whereby the immune headquarters produces antibodies, and these antibodies, you see these antibodies, the immune headquarters can produce antibodies, and these antibodies, they come and block this kind of what we call the nicotinic receptors. So now, if it blocks the nicotinic receptors, what happens? It means acetylcholine will not come and bind on these receptors. What's going to be happening? We shall not have the formation of what we call end plate potential. Okay, so now that kind of a condition whereby the antibodies are produced against its own structure or its own tissues, blocking the nicotinic receptors, leading into absence of what we call the end plate potential. This is what we call myasthenia gravis. Okay, so now our second condition is going to be what we call myasthenia gravis. Okay, and this condition is going to be the autoantibodies are going to be blocking the nicotinic receptors, blocking the binding of acetylcholine, and then of course we shall not have the influx of these sodium ions, which will lead into what we call absence of the end plate potential, and then of course muscles will not contract. And this one will also lead what we call paralysis and even also what we call muscle weakness. Okay. Then the third one is also another beautiful condition whereby antibodies, instead of blocking this kind of nicotinic receptors, they just come and block these channels here. Okay. You see these channels, they are what you call voltage gated calcium channels. If you block these channels, that means you're blocking the entrance of what we call calcium. Now, if you block calcium entry into this synaptic node, that means we shall not have the process of fusion of these vesicles containing neurotransmitters to power the content to the synaptic cleft. That means you're going to be having very, very, very minimal of the vesicles fusing with the presynaptic membrane to release acetylcholine into the synaptic cleft. What does this one mean? It means that if you have less of the acetylcholine released here, we shall have less of the stimulation of these nicotinic receptors, and we shall have less of the local depolarization, and then we shall not have what we call sufficient muscle contraction. So someone will have muscle weakness and then also what we call paralysis. Okay, so now that kind of condition whereby there is antibodies blocking these kind of voltage-gated calcium channels, that condition is what we call Lambert. Lambert-Eaton, myasthenia, myasthenic syndrome. Okay, okay. And then another one is that we have what we call organophosphate poisoning. And with organophosphate poisoning is super, super dangerous in a way that it will come and then block this enzyme. How do we call this enzyme? This enzyme is what we call acetylcholine sterase. It is responsible for breaking acetylcholine after the impulse has crossed. Okay. Now, if you block this enzyme, what does this one mean? We are going to be having now a lot of acetylcholine in this cleft. Okay. And if you have a lot of the concentration of acetylcholine in this cleft, it will lead into now continuous stimulations. Okay, continuous stimulations, and that kind of the continuous stimulation is super, super very dangerous in the way that it is going to be keeping the membrane potential towards threshold. Okay, and now when you keep a membrane towards threshold, this will lead into what we call the blockage of what we call the inactivation gate. Okay, and then the sodium voltage gated sodium channels can block completely, and this is something which is very, very, very dangerous, more especially in what we call hyperkalemia. Okay, now this kind of condition is going to be as a result of what we call organophosphate poisoning. Okay, what we call organophosphate poisoning. Okay, organophosphate poisoning. Okay, so now having a recap on this clinical correlation, what we call clinical correlation. Okay, clinical correlations. We have said that we have what we call botulinum toxins, which are going to be produced by the Clostridium botulism, which is going to be a gram-positive anaerobic bacteria releasing these kind of neurotoxic toxins. These toxins, they have the ability to come and break these snare proteins, okay? And when you break these snare proteins, you shall not have now the pulling ability of these vesicles to fuse the presynaptic membrane to release its content, acetylcholine, into the cleft, okay? So now that is what we call botulinum toxicity, which you may even call what we call botulism, okay? And then we said with the myasthenia gravis, we have autoantibodies which are going to be coming and attack nicotinic receptors, blocking the binding of acetylcholine. We shall not have now these gates opening, and then we shall not have the influx of sodium to cause the local end plate potential, which will lead into what we call muscle paralysis and then also muscle weakness. Okay, and then we said with the Lambert-Eaton myasthenic syndrome, here we are going to be having antibodies produced on this voltage-gated calcium channel. We shall not have influx of calcium, and if we don't have calcium, these vesicles will not fuse with the presynaptic membrane. We shall have less of the acetylcholine in the cleft, and then we shall have no end plate potential being generated. Okay, that is Lambert-Eaton myasthenic syndrome. And then we said organophosphate poisoning. With organophosphate poisoning, these chemicals are going to be blocking the activity of this enzyme called acetylcholinesterase. And then, of course, this enzyme breaks acetylcholine. If you block this enzyme, we shall have high levels of acetylcholine in the cleft. And if you have high levels of acetylcholine in the cleft, this will lead into what we call a continuous stimulation of this muscle fiber to keep the membrane potential towards threshold. It can lead into the inactivation of what we call the inactivation gates of what we call these sodium, voltage-gated sodium channels. This is such another beautiful thing, okay? So now this covers neuromuscular junction transmission with some little bit of the physiology. If you think whatever I've talked is making sense, please make sure you like the video, comment down in the comment section, and make sure you subscribe. Thank you very much for watching from the beginning up to the end. And it has been Tumnad as always. Until we meet next time.